Good morning, everyone. Thanks for coming back for day two. I think we had a great day yesterday. The presentations were superb across the board. Today, hopefully, will be equally superb. We have a half a day today, 2 sessions this morning. The first is diet and prebiotics, and the second is microbial-derived molecules. And again, welcome. Thanks for your attention yesterday. We look forward to great presentations and active discussion today. I'm going to turn it over to this morning's moderator, Joel Duray. Thanks, Joel.
Thank you, Gail. Good morning, everyone. It's a pleasure to welcome you for day 2 of GMFH 2023 in Prague. I'm Joël Doré. I'm a research director with INRAE in France, and one of the co-founders of GMFH that has been really a fantastic adventure for the past decade. I'm involved also in the editorial work of the digital platform gutmicrobiota4health.com, together with Tinkle in Barcelona and a panel of scientific experts and representatives of our sponsors. And I suggest that you do visit the platform and register for our monthly newsletter if you have not joined yet. So I'm sure you all enjoyed day 1, and I'm happy to open this session of day 2. And our first speaker is Maish Desai. Maish is group leader of the Eco-Immunology and Microbiome team at the Luxembourg Institute of Health. Maish's lab focuses on discerning how fiber-deprived gut microbiome impacts on our health and contributes to inflammatory disorders. His team aims to design dietary therapeutics to strengthen the mucosal barrier and improve human health. Maish, the floor is yours.
Thank you, Joel, for this kind introduction. Good morning, everyone. I would also like to thank the organizers for an invitation to this great meeting. Here are my disclosures, and none of these are related to the work I'm going to present today. I'm showing you here a fluorescence microscope image of a mouse colonic thin section that is stained with a anti-mucin antibody. You can see the mucus layer in green that separates the microbiota from the epithelium. You can also see goblet cells packed with mucin granules that are about to be secreted to contribute to the mucus barrier. This is important because bacteria that you see here as tiny blue dots constantly keep on feeding on the mucus barrier, and it is important that the host keeps on replenishing the barrier. Now, this mucus microbiota interaction is very important to drive intestinal homeostasis. And why is it so? To explain that, I will show you the structure of one of the dominant mucins in the gut. It's called mucin 2. Mucins are glycoproteins, meaning they are made up of glycans. So you see different glycans listed there and different shapes, and they are connected via various linkages. Any guess how many of these linkages can be cleaved off by commensal bacterial enzymes? Surprisingly, all of them can be cleaved by commensal bacterial enzymes. Now just imagine what would happen if the balance between bacterial mucus degradation and the host production is tipped towards bacterial mucus degradation. Our previously published work that was also highlighted as a cover story of the German magazine Der Spiegel showed that a dietary fiber-deprived gut microbiota degrades the colonic mucus barrier and enhances susceptibility to a mucosal pathogen. Now, $1 million question is, when we have such a leaky gut, what happens to other diseases, such as inflammatory bowel disease or food allergy? In my presentation, I'm going to focus on 2 diseases, IBD and food allergy, and I'm going to show you some mechanistic evidence of how fiber consumption or reduced fiber consumption is connected to these diseases. In the final part of my talk, I will show you a translational intervention study, fiber intervention study in healthy humans that is designed to identify biomarkers that can be eventually used as early warning signs of barrier dysfunction in various diseases. The first part of my talk will focus on IBD. We all know IBD is increasing and industrialization is blamed. When we talk about industrialization, diet plays an important role. So diets reduced in fiber and high in fat are often considered as a culprit. But again, how the reduced consumption of fiber is connected to IBD has been poorly understood. So in the first part of my talk, this project was carried out by a postdoc in my team, Microbiota. We hypothesized that mucin-degrading bacteria catalyze important disease-promoting immune pathways in IBD. In our published work, we have shown that although in the fiber-free fed mice, the mucus layer is reduced, in the absence of genetic predisposition, so in this case, presence of IL-10, there is no obvious colitis. But this would change if there is presence of genetic predisposition, so in this case, lack of IL-10, which is an immunoregulatory cytokine. And so we hypothesized that this would lead to severe forms of IBD. To test this hypothesis, we employed a synthetic human gut microbiota with each of these strains empirically characterized for their abilities to digest plant and animal-derived oligosaccharides and polysaccharides. Since this was part of a published work, I will not go into the details of the synthetic microbiota, but one thing I would like to highlight here is there are 4 mucin-degrading bacteria in this synthetic microbiota, marked here in blue, that we have tested for their abilities to grow on porcine gastric mucinoglycans. So in this study, we colonized IL-10 knockout germ-free mice with either a 14-member synthetic microbiota, which I will abbreviate as 14SM, or a 10-member synthetic microbiota, which I will abbreviate as 10SM. So these 10SM lack the mucin-degrading bacteria. We then bred these mice, so respective 14SM and 10SM, and then we had pups that were weaned on either a fiber-rich diet or a fiber-free diet. Note that our fiber-rich diet is basically a chow, a standard chow, and our fiber-free diet does not include any microbiota digestible polysaccharides. Our readouts included colitis monitoring, so weight, then immune cell profiling, fecal microbiota composition using phylotype-specific qPCR primers, and changes in the IgA production by the host and coating of bacteria by IgA. Here is the first data slide that I'm showing you. So this work is available as a preprint, and it's currently under review. On the right-hand side, I'm showing you microbial compositions in 2 diets. And as we have published previously, we could see increase in the mucin-degrading bacteria in the fiber-free fed mice, so those which are marked in dark red. We could also see reduction in the mucus layer. This is not shown here, but this was as we had previously published in wild-type mice. On the left-hand side, you see a weight change plot over time in the 2 groups of 14SM fed 2 diets. In the beginning, we did not see any difference in the weight, but you can see that after about 60 days, the fiber-free fed mice started to lose dramatic amounts of weight, and some of them even had to be euthanized because they reached the human endpoint. We then asked what happens in addition to the reduction in the mucus layer, what happens to the permeability of the mucus layer, which we determined using fluorescent beads, which are bacteria-sized, which is exemplified here with the cartoon shown on the left side. On the right-hand side, I'm showing you 2 plots. One is for mucus penetrability, and the other one is for the distance of bacteria from the epithelium. As you can see in these plots, the IL-10 deficient mice showed more penetration of mucus by bacteria as their distance from the epithelium was significantly reduced, which we did not see in the wild-type mice that were sufficient for IL-10. Now, what happens to those mice which lack the mucin-degrading bacteria? So I'm showing you on the right-hand side, again, the weight change for the last few days. And I'm including here now the data for the 10SM mice. And as you can see, it's a very nice data. The 10SM mice, despite being fed a fiber-free diet, they do not lose weight. Now, what can we conclude from this data? These results suggest that combined low dietary fiber and mucin-degrading gut bacteria cause lethal colitis in genetically susceptible mice. And this is driven by mucin foraging of the microbiota. We then asked, what are the underlying immune pathways that drive these phenotypes? Because having mucin-degrading bacteria or not having them and understanding the immune pathways, this is a perfect opportunity using this model. So we looked at immune cells for 2 time points, which are marked here, an earlier time point, day 35, and a later time point, day 77. So in the next couple of slides, I'm going to summarize the data for immune cells. Overall, we saw that fiber deprivation increases the recruitment of Th1 and Th17 cells in the colonic lamina propria. These are data just for the 14SM mice, so I'm showing you 2 microbiota combinations, 10SM, 14SM, for 2 days and 2 diets. And when we look at what happens to the 10-member microbiota, we see that there is reduced Th1 responses in the 10-member microbiota despite absence of fiber. Well, it's actually, there's no difference in the 10-member microbiota between the Th1 responses, and the Th17 responses are reduced as compared to those in the 14-member microbiota. In addition to the Th1, Th17 axis in colitis, IgA secretion by the host and its coating by bacteria is also considered as a hallmark. What we saw was that fiber deprivation and mucin foraging increased the cecal and fecal IgA, and the IgA-coated bacteria were also increased in the 14SM mice, but not in the 10SM mice, suggesting that mucin foraging in the context of fiber deprivation is essential to increase the IgA production and also coating. We then asked which of the bacteria have altered coating. So we looked at all our 14 members, and we found that Akkermansia muciniphila's coating was significantly reduced, despite its presence being more than double in the fiber-free conditions, which you see on the right plot in the fiber-free fed mice. What can we conclude from this data? These data suggest that some bacteria like Akkermansia muciniphila are able to escape coating of IgA, which is why they perhaps drive type 1 responses, and this is then joined by other pathobionts such as Escherichia coli, which also shows reduced coating, and all this contributes to the lethal colitis that we observe in IL-10 knockout mice. Now here's the summary for the first part of my talk. I've shown you how specific diet and gut microbial factors contribute to inflammatory bowel disease. In the context of fiber-free diet, we see increase in mucolytic bacteria, we see increase in mucus penetrability, we see increase in IgA-coated bacteria overall with reduced coating of some specific members, and there is a very strong Th1, Th17 inflammation with mucin foraging bacteria specifically driving type 1 responses. As I said, this work is available as a preprint and is currently under review. I'm now going to switch to the second part of my talk, which is focused on food allergy, which a project that was done by a PhD student in my lab, Amy Parrish. The current understanding of food allergy relies on the equilibrium between oral tolerance and its connection to tolerogenic mucus and beneficial microbiota, and at the same time, how this is connected to allergen sensitization, which is driven by the epithelial breakdown of the epithelial barrier, which then drives type 2 responses, which contain Th2 and ILC2 cells, which then coat the mast cells with specific IgE secreted for the specific allergen. The epithelial barrier can be also broken by other environmental factors such as allergen, protease activity, food additives, or detergents. And here we asked what would happen when we have increased mucin foraging, more mucolytic bacteria. We hypothesized that this might also be a trigger for barrier breakdown and breakdown of the oral tolerance. So to test this hypothesis, this time we used SPF mice. These are BALB/c SPF mice, and we sensitized them for O-albumin, which is a model of food allergy. I'm showing you this plot for the anaphylaxis score. Again, there are two groups, fiber-rich fed mice and the fiber-free fed mice, and we clearly see that the fiber-free fed mice containing a cholera toxin, which is CTX, they have significantly increased anaphylaxis symptom scores. We then looked at what happens to the microbiota. We noticed that just sensitization alone is able to drive some changes in the microbiota. And one of the key members of these changes was Akkermansia muciniphilia, which was, although increased in the fiber-free conditions, it was, after sensitization, almost completely lost, or in the fiber-rich conditions, but not in fiber-free conditions. So this suggested to us that Akkermansia is somehow playing a role in the sensitization process or driving some immune responses. Now, how would one test that? We then went back to our synthetic microbiota models. So we used our 14-member synthetic microbiota, and this time we only removed one specific member from the microbiota, so Akkermansia muciniphilia, to have a 13-member microbiota, and then we carried out the same food allergy model. So I'm showing you here the microbial composition. I will not go into the details of that, but I want to get on to the main results here. I'm showing you here again anaphylaxis score in the 14SM mice for two diets, again red and green colors, and we could clearly and very nicely recapitulate our SPF phenotype in the notobiotic settings, and we saw there was an increase in the anaphylaxis symptom scores in the fiber-free fed mice. And what happens with the 13SM mice lacking Akkermansia muciniphilia? We saw that there was no difference. So this diet-mediated difference was completely abrogated. And when we look at germ-free mice are known to have higher susceptibility to food allergy or any allergy because they are type 2 skewed. And so here we did not see any difference between the two diets, but the germ-free mice had higher anaphylaxis scores. But one thing that is important to notice here is that the moment we colonize with 14SM containing Akkermansia, in the context of fiber-rich diet, you see reduced symptoms, which highlights some potential beneficial effects of Akkermansia. But when we have a fiber-free diet in the 14SM, there's increased symptoms of anaphylaxis, which highlights potential detrimental effects of Akkermansia muciniphilia. We then wondered what sort of immune pathways are changed when we have Akkermansia, we don't have Akkermansia in the context of diet or in the absence of fiber. And to investigate that, we performed mass cytometry time of flight. So I'm showing you here data for immune cells. In general, we saw Akkermansia muciniphilia drives both diet-dependent and independent responses. So among adaptive cells, we saw that in general, having Akkermansia led to higher type 2 cells in both diets, and not having Akkermansia led to more type 1 cells in both diets. And among the type 2 innate cells, we saw that in general, Akkermansia and fiber deprivation increased the type 2 innate cells, and also eosinophils. One of the important findings of this study was that we saw there was higher coating of bacteria by IgE, not IgA this time, but IgE, and this effect was increased when Akkermansia muciniphilia was present. So all these data bring me to this model that we have put together. In general, in the fiber-rich conditions, we see an allergy response that is known in the textbook. So we see a type 2 response, we see increased mast cells, we see systemic IgE. But when we look at, and this is all further promoted by Akkermansia mucinephila, but when we look at what happens in the fiber-free conditions, which is shown here on the right, we see that these type 2 responses are further joined by some innate type 2 responses that are driven by eosinophils, CD8 positive cells that are producing IL-5, also type 1 cells, so Th1 NK cells. And we don't see systemic IgE. We rather see that bacteria get coated with IgE, so there's more local response of the IgE. Now, this model can be considered as a novel endotype of food allergy because the left-hand side is what would be detected with a skin prick test, but the right-hand side model is more a model of eosinophilic colitis, and in the absence of systemic IgE, one would not detect it with the skin prick test. This work is also available as a preprint and currently is under revision. I'm now moving, I will move on to the third part of my talk, which is an intervention study. How many minutes do I have? I don't see. 10 more, okay. It's an intervention study. So whenever I presented our data for the few fiber mucus axis, I was often asked, does this work in humans? And so that was a very good motivation to start this intervention study, which was done by a very talented PhD student, Erika Grant. And so we call this a Luxembourgish fiber cohort, or in short, LuxFICO. Now, fiber intervention, as we all know, has been done in humans, and there are several really good papers on this, so you might be wondering why, again, fiber intervention. But it's important to note that no studies have investigated how low fiber affects the abundance or activity of mucin-degrading bacteria, and specifically in a longitudinal study where there is an intervention of fiber. So this is our study design. We had about a 5-week intervention, and the key strengths of this study is that this is a crossover study design, meaning that participants serve as their own controls. And these are controlled diet interventions, so participants get 3 meals a day. These are freshly prepared meals. They're delivered to their work or home places. And at the baseline, their food frequency questionnaires and all the data are recorded. And the goal here is to identify diet-responsive microbial biomarkers that can be used as early warning signs of barrier disruption. Now, just to give you a perspective, I'm showing you what are the recommendations of the fiber recommendations of for the European countries. And you can see that the high fiber recommendations are, so our fibers interventions were 40 gram for high fiber and 13 gram for low fiber, and you see the adequate intake is 25 gram per day that is recommended, and then you see it also for various different European countries, how they are below the adequate intake. On the left-hand side, you see that there is no difference in the calories. These are both healthy diets, but there's only difference is the amount of fiber that is in gram per day. And so the participants underwent this intervention for 1 week, and then there was a washout, and then another intervention, so randomization, and then again a washout, and then we collected stool samples at the end of each intervention or washout. So just to give you a perspective, what is 40 gram of fiber and what is 13 gram of fiber? So basically 40 gram of fiber is 10 apples a day, and the low fiber diet is equivalent to 3.5 apples a day. The participants were all metabolically healthy. They were living in Luxembourg for the past 2 years, but they have various genetic backgrounds, as you can see on this map. They come from various countries, and they are relatively younger, and most of them are females. And so very quickly, I will not go into the details of this data, but we saw that there were subtle improvements in metabolic markers on both diet interventions. And this could be because both diets are healthy. They don't contain any processed foods. And we also saw an increase in the CRP marker, which is a marker for inflammation. So it's interesting to go into the details of individual responses, why we see such kind of inflammatory markers. In the interest of time, I will only focus on 2 readouts. So we are looking at various different readouts. It's still an ongoing study. We have enrolled 30 participants. So I will show you data for 16S sequencing and glycan degrading enzyme activities. So the very first, I'm showing you this PCOA plot. It's colored by, so there are 359 samples from 30 donors. This is colored by visit type or donor. So you see different intervention arms there. And the important thing to note here is that there is inter-individual variation, which is not surprising. We all know that. But this is where our study is important, because we can focus on we can ignore this noise of inter-individual variation, and we can focus on how intervention in a particular individual can give us some novel insights. So we saw that several taxa were enriched in the fiber-rich conditions or on the fiber-free conditions. This is a very busy slide, but bear with me, I will walk you through it. Basically, what I'm showing you here is a fold change fiber-rich, fiber-free. Then you see different phyla that are marked in different colors. There is a different study arm shown with a dendrogram at the top of the heat map. And that shows basically, it doesn't matter which study arm we are considering, the composition of the microbiota is, the effects are the same. And then we see the intervention again, high fiber red, low fiber in high fiber green, low fiber red. And so it's important to note here, and so we also see that they are listed with the log Q values, which you see on the right-hand side, and the very right is the list of the genera. On the left-hand side, you see the prevalence, meaning in how many individuals these taxa were present, and then I'm also showing you the abundance of these taxa. So we saw that there were certain bacteria increased in the fiber-rich fed participants, and these were 5 genera. And I'm going to highlight 2 of those, which are Eubacterium ventriosum and Agathobacter. And these we know are potent fiber degraders from our in vitro growth assays. And it is interesting to note that in our mouse studies, this bacterium also virtually disappears. So it was nice to see similar effects in humans. Now, coming back to our original idea, what happens to the mucin foraging, we saw that there was a very potent mucin degrader, Ruminococcus torquis, that was increased in several participants that were fed a low-fiber diet. In addition to that, so these are data for Ruminococcus torquis. In addition to that, we also saw that some other taxa were increased on the fiber-free diet. But we wanted to look at what happens to the enzyme activities of these bacteria, because we are at the moment culturing these mucin-degrading bacteria, but in the meantime, this gives us a very good idea about what happens to the mucin foraging in the absence of fiber. So on the left-hand side, I'm showing you comparison based with the baseline data, and you see clearly that 2 enzymes which are known to degrade linkages in the mucin, fucosidase and N-acetylglucosaminidase, they were increased in the fiber-free fed individuals. And on the right-hand side, I'm showing you data for direct comparison where we saw increase in sulfatases. So sulfatases are enzymes that cleave off sulfate groups from mucins, and without cleaving off these sulfate groups, bacteria cannot feed on the other structure of the mucus. So this is a very important piece of data. So just very quickly, we also saw some other pathobionts that were increased under low fiber conditions, which may not be all mucin foragers, but they might be related. For example, we saw increase in subdilogranulum, which was increased, which is associated with IBD and rheumatoid arthritis, or also Collinsella, and also some other pathobionts. So the goal here is that we would like to culture these bacteria and then design dietary therapeutics in order to keep them on the commensal spectrum of the microbiome so that they don't become pathobionts. So in summary, I've shown you that we could recapitulate the findings in mouse models, and when some of the things now, there are new results in the human studies, I get often asked, does it work in mice? So we are going back to the mouse models, and we are looking at some of the specific effects of these bacteria. And then we also saw that there was an increase in enzyme activities, which could be a proxy for barrier disruption. And as I said, the next steps are to isolate and characterize these microbes. So this is my final data slide. I'm showing you, this is another study in the lab. It is also available as a preprint, just to highlight that it's not just fiber-mucus connection that drives the effects on the host, but only the metabolism of fiber without the mucus could be also important. For example, in this study, we show that dietary fibers act as precursors for B vitamin synthesis. And so when there are, this is a mouse-only study, and I'm showing you example for riboflavin there. And so you can see that the fiber-rich fat groups, I will not go into the details of that, they all have higher levels of riboflavin. It's the same for several other B vitamins, and these are reduced in the fiber-free conditions, and we almost don't see any of these in germ-free mice, which suggests that these are produced by the microbiota. And so in the fiber-free conditions, we see that there is actually more utilization of the B vitamins, and in the fiber-rich conditions, more is available to the host. So this is my very final slide, which I will use to both summarize my talk and give a perspective of my talk. And this is from an article that my lab published the year before, where we use the term microbiome engineering. So our goal is that we would like to use different biomarkers that we are identifying in order to boost the existing therapies. For example, we are also working on IBD cohorts in relapse and remission, and so we are looking at what happens when there is a relapse or remission to the mucin foraging, and when they're undergoing antibody-based therapies, how we can harness the effects of diet, and we can supplement this with the existing therapies. And for that, we are using several tools, such as personalized nutrition, randomized controlled trials, as one I just explained. And then one of our very important tools is to go back to notobiotic models, and all this, we hope, can help us in engineering the gut microbiome. And with this, I would like to end and thank several of our very talented PhD students and postdocs, our collaborators all over the world, the funding agencies, and you for your time and interest. Thank you.
Thank you, Mahesh. I should have given you 2 minutes when you asked. We are going over time, actually, so I will not open to discussion, but please, everyone, grab Mahesh during the break.
Okay.
He will be around and happy to answer.
Thank you. I couldn't have a track of time.
Thank you very much. Thank you. I will introduce the next speaker. The presentation will be given by Matthew Carter. Matt is a member of Justin Sonnenberg's laboratory in Stanford University in the Department of Microbiology and Immunology. His research explores the avenues of communication between diet, gut microbiota, host metabolism, and immunity. Before Stanford, Matt worked with the company Caribou Bioscience in Berkeley, California. Matt, the floor is yours.
Thank you for the introduction. Thank you to the organizers for inviting me. And also thank you to both the previous speaker and also Professor Madsen yesterday for giving such a wonderful introduction to prebiotics and dietary fiber. Today I'll be talking about a class of prebiotics that we haven't really talked about so far, and that is human milk oligosaccharides, or HMOs. So HMOs are the second most abundant component of human breast milk after lactose. And they really are a beautiful example of host microbe coevolution. They are synthesized by the mother, and they are delivered to the infant via breast milk, where the infant cannot metabolize them directly for energy. Rather, they are sort of designed to feed the nascent infant gut microbiota. And more specifically, there are sort of specialist degraders, such as bifidobacterium, that are among the first colonizers. And sort of by feeding these bifidobacterium and other bugs, it sort of helps to foster a healthy trajectory of the gut microbiota in infants. This results in production of beneficial metabolites, such as short-chain fatty acids, colonization resistance against pathogens. This also helps to train the developing immune system, and also in clinical trials looking at formula-fed infants with and without HMO supplementation, it's also been shown that HMOs are associated with proper cognitive development of infants. Where the study that I'll be talking about today takes a left turn is that we took a purified human milk oligosaccharide called 2-fucosyl-lactose and gave it to a cohort of adults, more specifically a cohort of adults age 60 and older. We call this the RAMP study. I did not come up with the acronym. This was a study led by my advisor, Justin Sonnenberg, and our collaborator, Christopher Gardner, in the Department of Nutrition at Stanford, and it was funded by Abbott Nutrition. And so just to give some explanation of why we decided to give an HMO to a cohort of adults, it really has to do with the fact that, that both our microbiota and our immune systems change as we age. So as we age, our sort of alpha diversity and alpha diversity and richness decreases over time. This is associated with increased susceptibility to pathogens, also a changing sort of metabolomic profile of the gut, namely a decrease in short-chain fatty acids and changes to circulating metabolites. And you'll notice that some of these changes that you see in the aging microbiota are kind of the opposite of what you see in the developing gut microbiota. So it's not that they're perfect mirror images, but I think the analogy is interesting. With respect to the immune system, this is some amazing work from Mark Davis's lab at Stanford comparing the immune system of a young cohort and an older cohort, where what they did was they isolated immune cell subsets from these subjects and then stimulated these immune cell subsets with a panel of different cytokines. And so in the panel on the left, you can see a sort of representative young person. Here is CD8 T cells stimulated with IL-6, and you see a nice dose response curve through. I should have mentioned that the output they were looking for is signaling through the JAK-STAT pathway. And so you see this nice dose response curve in this young person, whereas in the older person, you see it doesn't really matter how much IL-6 you give to these T cells, there's no response through STAT3. So you have this sort of hypoactivity in an older immune system. In addition to that, if you look at the unstimulated condition specifically, you see that there is also a heightened level of baseline inflammation in the older person. And so the authors looked across many immune cell subsets and cytokine stimulations and saw that this was kind of a reproducible phenomenon. And so what the authors did was, from this data, they derived what they called a cytokine response score, basically a measure of how responsive one's immune system is to stimulus. And this was positively associated with cardiovascular biomarkers. So a higher cytokine response score was better. And so with these observations to the microbiota of the immune system, as we age, we thought that a sort of a prebiotic that is known to stimulate both the microbiota and the immune system would be an interesting intervention to do. And so here is an overview of the trial design. There was 3 treatment arms. There was a placebo group, a low-dose 2FL group consuming 1 gram of 2FL per day, a high-dose 2FL group consuming 5 grams per day. Really the only inclusion criteria was being 60 or older, and the exclusion criteria sort of excluded anyone with metabolic syndrome or some history of gastrointestinal disease. It was a 6-week intervention. We took 2 baseline time points, a midpoint time point at week 3. In addition to the end of intervention time point at week 6, we took a washout time point 4 weeks after cessation of consumption of 2FL or the placebo. And we collected urine, blood, and stool to analyze the microbiome and also perform metabolomics and immune profiling on the blood. And so the primary outcome that we pre-registered for this clinical trial was that cytokine response score that I mentioned from that previous paper. And unfortunately, there was no change over the course of the intervention in any of the treatment groups. We hypothesized that you would see an increase in the 2FL consuming groups. So it goes with doing human studies, but plenty of secondary outcomes to go through. So one of the things that we looked at was we performed targeted metabolomics of blood and urine, specifically looking for 2FL in these matrices. And indeed, we do detect 2FL in appreciable quantities, especially in the high-dose group. And so on the one hand, this is a measure of adherence to the study protocol because we can see this in circulation. And because we don't see it in the placebo group, really, we know that it is not some sort of baseline component of people's diet. We performed a standard sort of lipid and metabolic panel on these individuals, and over the course of the study from baseline to end, there were 2 features that changed significantly, both in the high-dose group, and that was fasting insulin and HDL cholesterol. So HDL cholesterol, good cholesterol, that was encouraging. The insulin finding, I think, was surprising. Naively, I would associate higher insulin with lower insulin sensitivity, but it is worth noting that we did not see changes in fasting glucose, and also people's weight did not change over the course of the study, indicating no real change in adiposity. Now looking at the microbiome, this is 16S sequencing data. We didn't see any changes in alpha diversity over the course of the intervention. When we look at beta diversity, overall composition, we see that specifically at the week 3 midpoint, there is a significant variation in composition, and this is driven namely by the difference between the high dose and the placebo group. And interestingly, this difference in composition subsides by the end of the intervention, or at least is not significant anymore. And when we drill down to specific taxonomic units that change during the course of the intervention, it is exactly what we expected to see, which is that the sort of specialist degraders of HMOs, Bifidobacterium, increased in the high dose group to a lesser degree in the low dose group. And so you can see that the high dose group at week 3 is significantly greater both than the baseline time points and compared to the washout, indicating that when you remove this stimulus, Bifidobacterium subsides back to baseline. So you get a rather specific and reversible modulation of the gut microbiota. It is also interesting to note that for some people, Bifidobacterium does decrease in abundance even at the week 6 time point, which is somewhat consistent with what people observe with sort of resilience or some homeostatic mechanism in the ecology of the gut. And so we also performed untargeted metabolomics and targeted proteomics of serum. The targeted proteomics approach that we used was this platform called O-Link, which is basically a panel of 92, mostly cytokines, but also a few hormones. And what we observed was a decrease in octanoyl carnitine in the high dose group and an increase in the hormone FGF21. I'm not an endocrinologist, but from my read, FGF21 is associated with stimulating insulin secretion, which perhaps meshes with the heightened insulin levels we see in the high dose group. There have been clinical trials looking at agonists of the FGF21 receptor, and these participants in these clinical trials also see higher levels of HDL cholesterol and sort of depressed lipid metabolism. And so while I'm not suggesting sort of what the mechanism of this might be, I think there is sort of some coherence to the data that we're seeing. And so one thing that sort of commonly happens when you're doing these studies in humans is that your results are hugely influenced by people's gut microbiome composition at the start of the intervention, and that you see large amounts of variation between people. Specifically for this study, because a bloom of Bifidobacterium was kind of the main microbiome finding, we wanted to kind of subset this data in a different way because some people simply don't have Bifidobacterium in their gut microbiota, and so if it's not there, then it can't bloom. And so what we did was subset individuals into a Bifidobacterium responder or non-responder on the basis of this change in Bifidobacterium abundance from week 0 to week 3. And so it's basically everyone in the 50th percentile and above. And when you look at the abundance of Bifidobacterium at baseline in non-responders and responders, you see that responders do have more Bifidobacterium at baseline, although it is not statistically significant, driven by those couple of outliers. And we also see that Bifidobacterium responders also have more 2FL in circulation at week 6. And so now when we subset the metabolomics and the cytokine or proteomics data in light of this non-responder-responder subsetting, we see sort of additional metabolites either associated with Bifidobacterium bloom or negatively associated, and these are mostly amino acids that seem to change in circulation. The cytokine data looks kind of strange. In this panel of 92 different cytokines and hormones, most of them are higher in the responders. Interestingly, if you look at different ways of subsetting people, even just based on like the high dose group versus the placebo group, it looks like a totally normal volcano plot, and it really is only when you subset individuals in this fashion that you see this result. Because there's only 92 cytokines in our panel, it's kind of challenging to do standard sort of gene set enrichment or pathway analysis because you're not looking at the entire proteome in an unbiased way, but it seems that some of these cytokines are enriched for chemotaxis of immune cells and also response to organic molecules. And so The last thing that I'll show is basically looking at a correlation network between the metabolomics data and this proteomics data, where obviously within each data set, one would expect metabolites to be correlated with one another, or cytokines reflecting metabolic networks or regulation networks. But here we were particularly interested in the metabolites and cytokines that were associated with one another, so kind of between these data sets. And this just kind of pulls out what those specific associations are. And so some of these I think that I'm interested in are some of these metabolites were associated with the bifidobacterium responders, namely like taurine and ornithine here. So obviously, again, this data is sort of associations, but these are some interesting hits to follow up on. Okay, so to conclude what I've told you about the RAMP study, this was a clinical trial in individuals aged 60 and older where we gave people 5, 1, or no grams of 2FL per day. We observed 2FL in circulation in these people, and we saw a bloom of bifidobacterium that was reversible upon removing 2FL from the diet. And this was associated with an increase in HDL cholesterol and fasting insulin. And when we performed this subsetting individuals based on bifidobacterium response status, we saw sort of additional cytokines that were associated with responders, as well as changes to circulating metabolite pools. So I want to conclude by thanking my lab and my advisor, Justin, who passed his invite to this conference on to me, as well as our collaborators in the Gardner Lab at Stanford, as well as the Stanford Human Immune Monitoring Center, and our scientific collaborators and funders at Abbott. Thank you for your attention.
Thank you. Thank you very much. It's open for questions. We have questions here on the first row, please.
Excellent presentation and idea. Would you think that there would be genetic predisposition of those responders and non-responders then would make these changes?
Genetic predisposition? That would be associated with whether or not they have bifidobacterium to begin with. That's an interesting question. I think we do generally tend to acquire bifidobacterium when we are infants, so I wonder if it would be more of an acquisition process when they were young that they carried through. I'm not sure about a genetic predisposition, but that's interesting.
Hi.
Oh, yeah, we have a question at the back there.
Yes, hi, very interesting. Do you have strain level information of bifidobacterium in particular? It's been shown in animal models that different strains can have different effects of diet-induced obesity, for example.
Yeah, so it was mostly, with regards to the specific species, it was mostly Bifidobacterium adolescentis was the most prevalent, and that was, had the strongest signal with regards to that bloom. And there was also Bifidobacterium continuatum, were the two dominant strains that had a sort of like 2FL specific response.
Yes, we have another question at the back.
So, Yong-Sung from Korea. You did a cognitive test, so is there any change in cognitive function of participant?
Oh, sorry, can you repeat that?
Is there any change in cognitive function?
Oh, so interestingly, yes, we did do like functional profiling of the like metagenomics, if that's what you're asking. And there really wasn't any significant changes, at least nothing that kind of survived multiple hypothesis testing correction. And really, I think that's because the change to the microbiota was so specific that it wasn't sort of wholesale changes to metabolic pathways. It was just bifidobacterium that were responding to 2FL.
Yes, Marion is coming. Thank you.
Hi, it's great to see this data. It's really amazing, super interesting. I have several questions, but following up from the previous question, adolescentis is really very interesting and known from having a lot of production of GABA, and you were just showing the amino acid, but you think you have more than just the glutamate that you were showing?
Like were there changes beyond? Yeah, I mean, it's definitely the pipeline that we were using. It was, I think, a total of like 100 metabolites that were recovered in the library. So I think if we had broader coverage with a different pipeline, I definitely think there would be
Yeah,
more.
that would be super interesting. And then
Yeah.
another question. So you explained the overall goal of the project, but is there something also about longevity and then kind of like immunosenescence and other markers of longevity? Because it's a very exciting topic.
Yeah, I mean, I think it would be hard to make claims about longevity. Like, it's very hard. Yeah, it would require, I think, much longer, like longitudinal trials. But it is interesting. Certainly, there are like particular taxa that are associated with longevity, and I think Bifidobacterium is one of them. But mechanistically, I'm not sure what would be going on.
I'll have a question.
Okay.
Thanks for the great presentation. I'm used to Justin's work moving more towards a high diversity of fibers. So here you are offering a line where you have one type of fiber and normally promoting one taxon of bacteria. So what do you think in terms of promoting an impact on immunosenescence about just these two options, going for one type of fiber and bifidobacteria versus a rather general increase in biodiversity of the microbiota?
Well, I think that certainly, and actually the previous speaker cited the paper in his presentation, one of the, like a high fiber intervention study that we did in the lab. It actually wasn't, it didn't seem to increase in a change in diversity. And really, I think that is because when you have a sort of complex fiber mix that you're supplementing, you're kind of subject to the heterogeneity of people's gut microbiota to begin with. And so I think that is kind of one of the advantages of this sort of specific fiber modulation, is you kind of maybe have a stronger expectation of what will happen. With regards to the immunogenicity, it's hard to say. Certainly, like Bifidobacterium is obviously highly studied as like a probiotic. And with regards to its influence on the immune system in infants, Ram Xavier and Tommy Vatanen have done some amazing work looking at like immunogenicity of Bifidobacterium in the infant gut. Unfortunately, the data that I showed with like the cytokine response score, like the immune cell signaling, like was a null finding for this trial. So it's hard to.
And did you look at the short-chain fatty acids?
We looked at short-chain fatty acid. There was no changes across any of the sort of major short-chain fatty acids. I would say that in my experience working with these studies, especially in healthy people who are not dysbiotic to begin with, often I don't really see changes in short-chain fatty acids. I think if you had like mucosal sampling or biopsies, maybe that would be more informative.
Yeah, we have one question here up front. Thanks, Balfour.
So I'm Sartor, University of North Carolina. I'm interested, do you think in an adult diet, diverse diet, there are components that may have similar metabolic activities that would lead to persistence of these particular Bifidobacteria adolescens, et cetera, that probably initially were colonized in the presence of the human oligosaccharides?
Yeah, that's a great question. I'm not sure. I mean, certainly they are, Bifidobacterium are fiber fermenters. I'm not sure about like other specific fibers. You're talking about like maybe using the HMO to like cause this bloom and then some other fiber to like cause persistence of that bloom?
Exactly.
Yeah, that's a good question. I mean, certainly there have been studies looking at like, obviously, just probiotic delivery of these Bifidobacterium, and that seems to be dependent upon like the background state of the microbiota. And aside from like HMO interventions in adults, I'm not sure that I've seen people specifically try to like cause this persistence of Bifidobacterium through.
And a corollary of that is, did you try to separate your responders, non-responders to breastfed versus, maybe as an adult may not be able to recall that.
We don't have that information, but we're actually trying to track that down with our participants. We didn't ask them that in the initial screening, but I think it would be super interesting to find that out.
other elements of birth could be relevant. Really, yeah, good idea. Any more questions? So I think I will thank you very much for the presentation. Thank you. Thank you much. And I will open the floor to Yael Haberman. Yael is a MD, PhD, clinician scientist. She's a pediatric gastroenterologist at the Sheba Medical Center and head of the Sheba Microbiome Center. Yael applies data science to large clinical data sets combining diet, omics, and microbiome to better understand pathogenesis, build predictive models, identify new targets for intervention with a focus on IBD. Yael, the floor is yours.
Thank you. Thanks to the organizers for inviting me. It has been a great meeting. I tweaked the title a little bit to using omics to elucidate interactions between diet exposure, bacteria, metabolism, and the host gut mucosa. I have no conflict of interest related to these presentations. So, as suggested here, I am a pediatrician and a pediatric gastroenterologist, and I'm still amazed by the fact that we see this increase in several diseases, metabolic and immune-mediated, in industrialized countries. And this rapid changes probably tells us something about the etiology of those diseases relating to environmental, dietary, and not so much changes in genetic, because genetic doesn't change in such a rapid pace. So we heard about obesity, but it's not really just obesity, and we heard about allergic and asthma. Eosinophilic esophagitis is another diet allergic-related entity that has been tremendously increased in the last previous years. Celiac, autism, and Crohn's disease. And at least, some of those diseases, we know that diet and exposure play a role in the pathogenesis. I really like this review because it kind of simplifies the explanation why diet is linked to chronic disease risk. Healthy diet maintain a better mucosal layer, higher mucus, as we heard in the morning, while non-healthy diets affect the mucosal barrier, cause the leaky gut, and then less constrain the immune response. But also in the same review, they say that there is a complex interaction between the different variables. So diet can affect directly the host, can affect the gut microbes can affect the host can affect the microbes. So it's really an ongoing and complex interaction. And I think what we are trying to do with others in the field is to try to dissect this a little bit to understand the interactions and maybe to tell us something about causal effects, and then we take it back to the lab, look at mechanistic studies, and then go back to the human and design some more directed interventions. So a little bit of the things I'm going to cover in this talk. I will talk about these changes in early childhood that we heard about in different talks today and yesterday, and what I think we know, and I think what we still need to better understand. I will tell you a little bit about our attempt to define what is a healthy gut microbiome, looking basically at different diseases and comparing shared differences between different diseases and kind of highlighting more of what is an IBD-specific signal that we can maybe use as a biomarker or a target for intervention. And I will tell you a little bit about our unpublished data from the source cohort that combines diet, exposure, metabolites, and ileal mucosal transcriptomics that we try to put on a healthy Crohn's disease axis, and then to try then to manipulate this, or to try to understand how we can manipulate this in future studies to take the signal more toward the healthy signal and away from the Crohn's disease one. So we heard about this dynamics that takes place during our lifespan from childhood to adulthood and to longevity. A lot of things changes during that time, but we really understand that the first few years of life are the major impact, the major dynamics takes place there. This is showing the alpha diversity in our Israeli cohort throughout the years from childhood to adulthood. This is showing the taxa that are incorporated during the years of a lifespan, and this is a PICO-A colored by age. And it is really interesting because we still really don't know what pushes a baby, colored here in purple, to a yellow dot on the top or at the bottom, and really how this predisposes those children later on to disease X or disease Y, and how we can really implement better understanding of what caused this transition and how we can prevent some of those frequent diseases that we see during childhood. So this is in Israel. It has been shown all over the place for many years now in many publications, but we really don't still understand all the factors that affect this maturation. This study highlighted that diet, mother exposure, and some environmental factors are affecting this, but this only explains some of the variation in early life. This is another study that highlights more the role of industrialization along this line. So again, PC1 highlights the differences or the maturation by age, but PC2 is kind of more explaining the differences by industrialization. And then again, what was interesting is that the differences are not only witnessed at the community level, it's also in the type of the bifido and its ability to degrade the HMOs that other people before me just have talked and introduced. So as we go from more industrialized country, the type of bifido is less adequate in utilizing the HMO in comparison to the non-industrialized one. So this is interesting, and it's probably also maybe related to the immune development later on. So this is an approach that we took in our cohort to try to understand the effect of attending daycare. So we wanted to see if this is a major influencer of the gut microbial maturation in the second and third year of life. So we analyzed data from children entering daycare, 4 different daycares around our hospital, and another group from taking care at home care. What we have shown that in this age group, daycare class and the daycare itself is a major determinant of the gut microbial composition, much more than breastfeeding and mode of delivery. And again, it's a different age group than was studied before. And when we compared 2 children in the same age from the same daycare versus those from different daycare, we have seen higher similarity between those that share the same daycare. So children from the same daycare were more similar to their peers than to other daycare. And when we compare the daycare children to the home care children, we have shown a distinct microbial composition and linking to a more mature microbial composition. So, of course, everybody asks me if this is a good thing or a bad thing, and I keep saying that in this research we don't know, but if we quote other papers that looked into this, they have shown that attending daycare is a protective for some of the allergic and asthmatic diseases. So this is a very nice initiative from Canada called the Child Cohort that is really looking into some of those things. They are following more than 3,000 children from birth to early childhood and are trying to link different exposure to microbial composition and then endpoint like asthma and allergy. And they have shown, for example, in this paper that if you have different factors and then that influence either a Bacteroides predominance or Proteobacteria predominance, this relates to later development of asthmatic and allergic diseases. And I really think that we need more such large cohort from around the world to better capture these different dietary and exposure to better understand this very crucial window of opportunity during childhood. Shifting to trying to identify what is a healthy gut microbiome. So when we were talking in different lab meetings, we keep saying that we see some of the same signature in different diseases, and we thought that some of the problem in doing this is because each cohort is looking at specific disease and a control group, and what we try to do is to try to compare between different diseases, and the approach we took is to look at to do an analytic meta-analysis. We aim to identify shared microbial responses between various diseases, also to identify an IBD-specific signal, and to use the non-disease-related signal to build basically a composite health index that can be calculated per sample and can tell us how healthy this is on this axis. So we use the public domain. This is a 16S analysis, 16S based analysis of 59 different disease cohorts, including almost 13,000 of samples from really many different diseases. And in this analysis, we compared each cohort within itself. So we characterized the effect size and the differences between each disease and control group, and then performed statistical tests to identify what is considered persistent signature within the different cohorts and what is kind of more specific to IBD. So this heat map summarized the different cohorts, and as you can see here, just by looking at it, there is a block of blue on the top and a block of red on the bottom, and this is something that is shared between the different diseases. Again, some discussion we had in the lab meeting is some people thought that maybe the bacteria are kind of a reporter of more of a disease state. Others thought that this may be just a bad habit or bad diet that is linked to some of those diseases epidemiology, but this is still an unknown. We don't know the answer, but this is, I think, is an interesting speculations. And here also we highlight the IBD-specific bacteria. So this is mainly of a block of this red in the middle that is increased in IBD and is kind of more of maybe of an inflamed bacteria composing of Gamella, Villionella, Fusobacteria, Streptococci, and those are enriched for salivary bacteria. So as I said, we are using this like more general microbial differences to compose a healthy index where we calculated the ones that are decreased divided by the ones that are increased, and then we have a per subject, per sample index. Shifting to the source cohort. So RISC is another cohort that I was heavily involved in. It's composed of 1,100 Crohn's disease patients. We used their transcriptomics, epigenetics, microbiome, and we really characterized how those relate to pathogenesis and how those are related to specific outcomes. But what we were missing in RISC back in the days was the dietary, environmental exposure, and metabolomics. And in SOURCE, we really wanted to capture these variables as well. And we wanted to explore, using diatomic, the interaction in the onset of Crohn's disease. So these are all newly diagnosed Crohn's disease, but also using a more heterogeneous population, using rural-urban West-East population from China, taking advantage a little bit of the changes that China is going through in the last decade, clinical studies. Just to summarize this in other work from other people, this is an interesting cohort from the Netherlands, looking at the output of plasma metabolome, looking into genetics, diet, microbiome. So what they concluded is that the diet and the microbiome are predominant factors linking the metabolome in the serum. But diet gave us the most numbers, the higher numbers of metabolites in the serum. And they also came up with different integration, mediation analysis to tell us something about causal interactions, but of course we all understand the need of interventional studies and functional studies to better understand this and go back to the patient cohorts. These are just examples of different in vitro studies and inpatient studies of different interventions. So this is the one example of using phytats to induce HDAC and enhance epithelial function. This is another study from the Sokol group looking at tryptophan metabolism, and this is also an interesting study looking at carboxymethylcellulose specific intervention in patients admitted to the hospital and were given a specific dietary modulation. These are all published data, but I think it just gives us this perspective of how we need to further look into this and go back again to the human cohorts. So just to summarize, I think there are many ways that we can probably better intervene, but we really need to understand how. We need to prioritize different metabolites, dietary factors, different intervention. We need to model, we need to test directionality and mechanism, and we need to go back to the patients and try them I really believe that childhood is a very, very important period. It's the window of opportunity to intervene and really need to understand better what's going on there. Want to thank my lab members, the collaborators, the fundings, and the Source Consortium.
Thank you. Thank you very much for this fantastic overview. We have questions in the room. I have one in the first row here.
That was fantastic, and I look forward to the publication of your source study. Many, many things you covered, but one practical implication I'd like to explore is the role of your association of iron with unhealthy microbiota. Did you distinguish between meat-based dietary iron versus supplemental iron given, and are there implications for an iron-deficient patient who needs iron but may have detrimental effects if given orally?
Yeah, so here we just capture the iron consumption based on the FFQ. So this included both food-based and supplements. We can go back and see, if we can retract this information. But we are also looking into we have a pediatric childhood cohort, and in Israel, iron is a mandatory or a suggestive supplement in early childhood between 4 months and 1 year. So we kind of asked ourselves really about the same question: is it really needed? Does it affect the microbiome in a specific way? And I think there we may be, because this was kind of more designed to answer this type of question, so there maybe we can potentially ask this question more directly, because this is what we designed to ask there. So I don't know the answer for what you were asking.
But I think I'm eager to see your results, because I think it's an extremely important therapeutic question.
I have one more question here. Two. Gary, you may start.
A really nice presentation. You showed fecal metabolomics. Did you do urinary or plasma metabolomics? The reason I'm asking is that there are really well-defined dietary biomarkers in plasma and urine. I'm wondering if there's an opportunity to sort of cross-validate your food frequency questionnaire with some metabolomics in either plasma or urine.
Yeah, so we do have the plasma, the serum metabolites as well. The signature was not so strong that correlated with the transcriptomic and the microbiome as we saw in the feces. But I think what you're suggesting is kind of checking our FFQ in the plasma, and I think this is something that we definitely can do. Thank you for that suggestion.
Yes, it's very fascinating. You have a lot of data. My primitive question is, since you made so many statistical correlation analysis, did you correct for multiple comparison? Because otherwise we might pitch the manganese issue, for instance, which might, I think, doesn't make much sense clinically, because we know that manganese may be reduced during intestinal inflammation, which is not a sign that it is the risk factor, but it's just the consequences of disease. So how can you distinguish between the most robust correlation you see and that could be some kind of random effect?
Yeah, so within each analysis we do correct for multiple comparison, but I understand that we do more than one analysis. So I think one thing here is that we analyze the Israeli and the Chinese cohorts kind of separately, and one cohort a little bit is a validation of the other cohort. So some of the thing we have seen to replicate in those two. The FFQs were not exactly the same because the population was not exactly the same, but I think what I'm alluding to is that I think really we don't like this so much in science, but in clinical related question, I think validation in independent cohorts is really the key. And I think this is what we are aiming to do and to try to validate the strongest. I think this will be the main part of the prioritization is really the validation.
One last quick question, Dan?
Congratulations. Very nice study. Looking to the study of the large database, when you look to the all diseases and all microbes, I don't know if I understood well, but it seemed that was a group of microorganisms in red that were more or less similar to all diseases. If this is true, this is the basis of your index. And if this is also true, could we use this for a clinical purpose to, let's say, screen populations or patients in a clinical way?
So I think you did understood right. There are many differences that are shared within different diseases, and we are using this in our different cohorts, different diseases, and it does hold up. Of course, there is overlap, right? If there are histograms, not all disease patients are here and all healthy patients are here, but there is a clear separation. And you know, in the discussion of this paper, we did suggest the use of this index for choosing FMT maybe, donors, or in different clinical trials, but we need to prove that this is working in that setting. But we did suggest this as an option there.
So we'll leave it here and move for coffee. I would like to thank the speakers for the great presentations with lots of engaging submitting and sometimes not yet published work, and the audience for the very stimulating discussions we've had. Thank you very much, and we meet again at 10:50.